Okay, so first things first, before I get into this, I want to just show my little card here with my name, my student ID, and my course ID. And before I get into the results of the lab, I just want to apologize for this assignment being late and the mess behind me. I've been absolutely slammed with work and school. I sent an email saying that I was going through finals, and that's no excuse for this assignment being late and my attention being so non-focused on this class. That's disrespectful to you, professor, and just to the school in general. I apologize for that. Um, I apologize. Let me get into the results of the lab. Let me start with plate number 1. So, plate number 1, you've got a really big let me see if I can try and get rid of the glare. So, you got really one big splotch of yeast there and some smaller ones around it. I couldn't call this one a viable plate because of the fact that I couldn't really count. I mean, you can count that one big splotch as one colony, and apart from that, there weren't very many other colonies, and it wasn't viable. I was really shocked by this because of dilution. I would have expected, if plate number 1 was not viable, I would have expected because it had more than 300 colonies and was therefore too numerous to count. Number 2, plate number 2. Apologies, let me find it real fast. Plate number 2. Plate number 2, if I recall correctly, was found to be viable. Now this plate, I I would expect the plate to be viable, but not for as low of a count as it had. Um, if I recall correctly, the count was somewhere in the 30s, so on the very low count of being viable, just really made the cut slightly, and that's not something I would have expected in the slightest. Uh, yeah, it really blew my expectations away. Now for number 3, this one, again, if I recall correctly, I believe it was considered viable. I don't have my data in front of me, but I believe that this plate was considered viable. And again, something I would have expected. I would have expected the colony count to be lower than number 2, and I don't believe it was. I don't believe it was. That is surprising because of the dilution. Logically, plate number 1 should have the most colonies, number 2 should have the second most, 3 should have the third most, and on and so forth. Now, number 4. Let me get into number 4. Number 4, I believe, was not viable, if I recall correctly, and it had a fair amount of growth. Now, this plate I would expect it to be on the verge of not being viable, and that's about what it was. It was in that 20 count and not quite there. So I wasn't shocked by the fact that plate number 4 wasn't viable. What I was shocked by was plate number 5. Now, plate number 5 had the most growth out of all the plates, and it was the quickest growing. The reason that this experiment took so long for me to upload was because no growth occurred for the first 48 hours, except for plate number 5. Plate number 5 had basically this exact same amount of growth through 48 hours as it did 72. I decided to wait a day to try and let the colonies show themselves a little more and have something to show for this wild failure of an experiment. Luckily, I did because I have something to show for it. If I had to call this experiment one thing, it'd be a successful failure. But yeah, so number 5, I believe it was not viable, and that to me that was expected. I didn't expect number 5 to have very many cultures at all, or colonies, I should say. Number 6, this one was not viable, and I was not shocked by that at all. I didn't expect number 6 to be viable in the slightest. I expected it to have maybe a single digit count of colonies because of the amount of dilution that occurred. That it had more than I expected. Not by a lot, but just a little bit. And that was a little shocking. Not shocking, but surprising. Surprising. That was a little bit of exaggerated use of words. But getting into the experiment as a whole, it did not go as planned because of the growth that occurred in the first 48 hours. Uh, Basically, no growth occurred, as I mentioned, other than plate number 5, which was really, it really scared me because they were all in the same environment. They were all exposed to the same amount of darkness, and they were in the same temperature, but only one of the plates were showing any sort of growth. And that was very, very concerning. So I decided to let it sit for another 40, not 48 hours, 24 hours, and it did show more growth. So I'm glad I did that. Um, but yeah, this experiment as a whole was really shocking to me. I didn't expect the growth to take as long as it did. One thing that I could perhaps say was a reason that the growth took longer was maybe the room was a little colder than it should have been, and for that reason the colonies took longer to form and the cultures took longer to show themselves. But I don't think that was the issue. I'm really at a loss for why this experiment took so long to show any sort of fruit. Another thing that could have possibly happened, I have no reason to believe that it happened or no proof that it happened, really just kind of looking back on the entire procedure and thinking what could have possibly gone wrong that could have caused all these issues, is that perhaps when I was pipetting the yeast mixture into the test tubes, there was a little bit of alcohol left in, and it killed some of the yeast. But that wouldn't make much sense considering it was only number 5 that showed growth in the expected period. That would mean that I'd have to have had alcohol in the pipette for 5 out of 6 of the dilutions. And I don't think that that would have happened. I was very careful in regards to that. So this is really just me spitballing as what could have possibly gone wrong. And yeah, I'm really sorry for this assignment being late. It won't happen again. I apologize for this whole thing. And to try and fix the problems that could have happened, I'm looking into buying an incubator to hopefully really control the climate that my cultures are in to make sure that they have the best possible chance to grow and the best possible chance of surviving. So thank you for uh, having grace and responding to my email. If I get points deducted, I completely understand. I obviously violated the late work policy, and it would be completely justified for you to knock off points from this video. But thank you for accepting it, and I'm very, very sorry. Please forgive me.